Yes, my dear students, today we will discuss the next topic that is known as the RNA world, ribonucleic acid. It was first considered as the genetic material. The second position have the OH group of ribonucleic acid, which make them more reactive, that makes RNA as a catalyst. It is evident that essential life processes such as metabolism, translation, splicing, etc. have evolved around RNA even before DNA has evolved as a genetic material. So we can't say that the RNA, it do not play any important role. Without RNA, the life process is impossible, like metabolism, translation, splicing, etc. Let us discuss what is the structure of the polynucleotide chain, that is known as the RNA. In RNA also, each nucleotide has 3 components as in DNA. As you have learnt, as the DNA, it possesses the nitrogenous bases are of 2 types, purine and the pyrimidines. Here also we can see that there are the purine and the pyrimidines are the 2 nitrogenous bases. Purine consists of the adenine and guanine, whereas the pyrimidine consists of the cytosine and uracil. Uracil means in place of thymine in RNA, it is known as the uracil. The sugar is ribose, which has an additional OH group on the second position. The nucleosides and nucleotides are called ribonucleosides and ribonucleotides respectively. Now, there are 3 types of RNA. One is known as a messenger RNA, second is known as a transfer RNA, and third one is known as a ribosomal RNA. What is the function of the messenger RNA? It brings the genetic information of DNA transcribed on it for protein synthesis. It is single-stranded, means all the information of the DNA, it simply transcribed into the messenger RNA. The transfer RNA, that is also known as a soluble RNA or the transfer RNA. The transfer RNA, it acts as an adapter molecules that reads the code on one hand and binds to the specific amino acids on the other hand. When we will see the diagram, you can understand. The transfer RNA has a clover leaf-like secondary structure, but actually it is an inverted L-shaped compact molecule. Let us see the diagram. Yes, it has an amino acid acceptor end, that is 3' end, and an anticodon loop where the 3 bases are complementary to the bases of the codon of the particular amino acids are present. Just you see in this diagram, it is the adapter molecules transfer RNA and as the adapter molecules. Here you see the codon arranged on the messenger RNA and the bases are the A, G, U. Its complementary base pairs are U, C, A. So at the anticodon loops, there are the base sequence which is just complementary to them, and the specific amino acids will bind to the 3' end. Third one is known as a ribosomal RNA. The ribosomal RNA, it forms the structure of the ribosomes. It also plays a catalytic role during translation. Means when there is the protein synthesis, when there is the formation of polypeptide chain, it plays very important role because it provides the site for the for the protein synthesis. So my dear students, up to here you have to learn and write it. The next topic which I am going to discuss, it will be the replication of DNA in my next video. Till then, bye.